Back to another session of biochemistry classes. So we finished with in the last class about the digestion and absorption of protein. So we'll go on further into the general metabolic pathway of the protein metabolism. So before we go on to the metabolic pathway as such, we should know certain reactions which are known as the general reactions of protein metabolism. So we all know that in the diet, the amino acids are obtained from the dietary proteins, right? So when we take in the dietary proteins, it enters into the amino acid pool, and we all have understood from the previous classes that unlike the lipids and the carbohydrates, the amino acids or the proteins are not stored in our body. So as we take in our diet, it has been utilized for certain other functions. So this we call it as a protein turnover. So what is it? The more important, the most of these amino acids are oxidized in the liver. So around 300 to 400 grams of proteins are synthesized and degraded. So the catabolism of the protein will lead to excessive loss of the nitrogen from the body. So we also have to know, depending on this, the catabolic pathways of all the amino acids. So we will be dealing only with the non-essential amino acids that are synthesized in our body. Okay, so now let us look into the amino acid metabolism as a whole. So when we talk about amino acid metabolism, we also have to know what are the sources of amino acid pool. So the dietary proteins, then the breakdown of the tissue proteins, and also the synthesis of the non-essential amino acids. So that's around 4 to 8 milligrams per deciliter. So now what is the fate of all these amino acids that are seen in the pool? It leads to the synthesis of the structural proteins, that is the tissue proteins, and then it leads to the functional protein synthesis like hemoglobin, myoglobin, proteins, hormones, and the enzymes, and also leads to the synthesis of small peptides, okay, like glutathione, endorphin, and enkephalin, and also leads to synthesis of certain non-protein nitrogen scoped like urea, uric acid, creatinine, and ammonia. And the catabolism of the amino acids gives to ammonia and alpha keto acids, right? So now when we look at the overall catabolism of the amino acids, it's an inter-organ relationship. So you can see in the kidney, the intestine, sorry, it is absorbed the amino acids and it is taken into the liver. Similarly, what happens from the intestines, the glutamate, it gets transported as alanine and citrulline and it enters into the liver. Now what happens in the liver? The toxic ammonia is trapped and it is synthesized as urea. And during certain conditions like starvation, the glucose is also synthesized from the amino acids, that is the glucogenic amino acids by gluconeogenesis. And then citrulline enters into the kidney. And then what is it? There is the branch chain amino acid that's also synthesized in the liver that enters into the muscle tissue, where again there is formation of glutamate. And the most important product of the ammonia, which is not toxic substance from the muscle, it gets transported in the form of alanine and it enters into the liver for the metabolism. Now from the muscle, the glutamate enters into the kidney and there it undergoes certain reactions, right? So what happens here? Ammonium is given off, glucose is also excreted, same way the arginine also. So this is the whole overview of the amino acid catabolism, right? So now when we look at the catabolism of amino acids, so what we all know is amino acid consists of an amino group and carboxylic group, right? So what is happening to the carboxylic group is removed as carbon dioxide by the decarboxylation reaction. Now the amino group is removed in the form of ammonia by a number of reactions, right? So we have first one is a transamination reaction by the enzyme aminotransferase, and we call it as transaminases. Okay, then we have the deamination reactions, which can be oxidative or non-oxidative. So oxidative deamination is by glutamate dehydrogenase or amino acid oxidase. And then you have one more, that is the non-oxidative deamination by amino acid dehydratase enzyme, right? So now what happens to the carbon skeletal of the amino acids? It leads to the formation of carbon dioxide and water by the TCA cycle, that is either by the glucogenic or the ketogenic amino acid. So now we'll go on to the first reaction, that is the decarboxylation reaction. That is nothing but the removal of the carbon dioxide from the free amino acid with the formation of the corresponding amines. So the amino acid, the presence of decarboxylase enzyme gets converted to the corresponding amine and carbon dioxide is given out. So the best example for this is histidine gets converted to histamine, tyrosine gets converted to tyramine. And we all know that from the previous classes that you've understood there are a lot of many biogenic amines which play an important role. So you know that you have dopamine, norepinephrine, epinephrine, tyramine, serotonin, melatonin, GABA, okay, then histamine, taurine and spermine are all the biogenic amines which are synthesized by the decarboxylation reaction, right? So next we'll go on to the transamination reaction, which is an important reaction which is frequently asked for you in the examination, and also it gives a clinical interpretation which is important for your case reports during your practical exams. So what do you understand by the name transamination reaction? It is a transfer of an amino group from one amino acid to keto acid with the formation of a new amino acid and a keto acid. So this reaction takes place in the liver. So all the amino acid except threonine, lysine, proline and hydroxyproline does not undergo transamination, right? So the keto acids involved are oxaloacetate, pyruvate and alpha ketoglutarate. So what is it? Transfer of an amino group from one amino acid to keto acid with the formation of a new amino acid and a keto acid. So we'll look into the examples of transamination reaction. So this is a substrate, that is the amino acid or the amino group donor. So now this gets converted to form a product of a keto acid. So here pyridoxal phosphate, PLP, is a coenzyme. This accepts the amino group to form the pyridoxamine phosphate, and this amino group gets transferred to form the product, that is the new amino acid, and the substrate, the keto acid, is an amino acceptor. So this is the basis of the transamination reaction. So we look into the examples of these. So there is two important enzymes that we know in transamination, that is serum glutamate pyruvate transaminase and serum glutamate oxaloacetate transaminase enzyme. So what is it here? Alanine plus alpha ketoglutarate gets converted to pyruvic acid and glutamate. So glutamate traps the ammonia group, right? So another enzyme is aspartate plus alpha ketoglutarate, the presence of serum glutamate oxaloacetate transaminase, and the coenzyme required here is PLP, gets converted to oxaloacetate and glutamate. Okay, so these two reactions you have to write as examples for transamination reaction. Now when you write about these two enzymes, you should also know what is the clinical importance of these enzymes, right? So what is it? Alanine amino transferase or ALT or SGPT levels are increased in the serum in liver diseases, example like infective hepatitis due to damage to the liver. And aspartate amino transferase AST or SGOT levels are increased in the serum in MI or myocardial infarction due to damage to the cardiac muscle. Okay, so now what is the other importance of this transamination reaction is the process involves a non-essential amino acids can be synthesized by this reaction. Okay, so this is about transamination reaction. So next we'll go on to the deamination reaction. So what is the deamination? That is the removal of the alpha amino group from the amino acids and ammonia is released. So here you have two types of oxidative and non-oxidative deamination. So in oxidative deamination, it takes place with an enzyme by amino acid oxidase enzyme. So this takes place in the liver and in the kidney. So what is it? It acts in the presence of water and produces ammonia and keto acids. So oxidases are of two types, that is the D-amino acid oxidases and L-amino acid oxidases. So when we say D-amino acid oxidases, it is present in the plants and in the microorganisms. The L-amino acid oxidases is present in the kidney and in the liver, and it requires the coenzyme flavin mononucleotide or FMN. So we look into one of the examples, oxidative deamination of amino acids. So L-amino acid, in the presence of L-amino acid, oxidase gets converted to alpha keto acid plus ammonia. So FMN is a coenzyme, gets converted to FMNH2 and hydrogen peroxide and oxygen is synthesized and one water molecule is released in the presence of a catalase enzyme. So this is about the deamination of amino acid. So next we'll go on to the non-oxidative deamination. So amino acids containing the hydroxyl group undergoes the direct deamination by the hydratase. Coenzyme required here is PLP. So the sulfur containing amino acids also undergo deamination by disulfide hydratase enzyme. So the hydroxyl containing amino acids are serine, threonine and homoserine. Okay, in the presence of dehydratase releases one ammonia, gets converted to the respective alpha keto acid. So sulfur containing amino acids cysteine in the presence of desulfur hydrase ammonia and hydrogen sulfide is released to get converted to pyruvate. So histidine in the presence of histidase, one molecule of ammonia is released, gets converted to uroconate. Okay, so this is the non-oxidative deamination. So next we'll go on to the oxidative deamination by glutamate dehydrogenase enzyme. So this enzyme is present in high concentration in the mitochondria of the liver and in the kidney. And this is an enzyme which requires NAD and NADP as a coenzyme. So glutamate is effectively deaminated to form the alpha ketoglutarate and free ammonia, and the free ammonia is highly toxic. So we'll go on to the reaction. So glutamate, in the presence of glutamate dehydrogenase, ammonia is given off, NADP gets converted to NADPH and gives alpha ketoglutarate, right? So next we'll go on to another reaction, that is transmethylation reaction. You would have studied this reaction, okay, in the vitamin chapter. So what is the transmethylation? How do you define it? It is the transfer of the methyl group to the acceptor molecule. So the compounds formed are choline, creatine, betaine, carnitine, epinephrine, melatonin and a certain other products are formed. Okay, so the dietary methionine is a major source of the methyl group. Okay, so what happens? The methyl group of methionine becomes available for transmethylation reaction only in an active form of methionine, and this active form of methionine we call it as SAM, or we say it as S-adenosyl methionine. Okay, so methionine itself is not a methyl donor. Only when it gets converted to S-adenosyl methionine gets converted to S-adenosyl methionine, where it gives the methyl group as a donor. Okay, so we look at the reaction, how it forms. So here you can see the methionine, okay, in the presence of methioadenosyl transferase enzyme where ATP is utilized, gets converted to S-adenosyl methionine, and then it is a methyl donor. So in the presence of methyl transferase enzyme leads to the formation of a number of products, we said compounds, non-epinephrine nucleotides, phosphodiene, phosphatidylcholine, right? And then it leads to synthesis of melatonin, okay? And then the guanidinoacetoacetate gives synthesis of creatine, betaine to choline, okay? So these are all accepting the methyl group and it gets converted to S-adenosyl homocysteine. And this S-adenosyl homocysteine gives off one molecule of adenosine to form the homocysteine. This in the presence of tetrahydrofolate where a methyl group N5 methyl tetrahydrofolate gets converted in the presence of vitamin B12 gets converted to methionine back, okay. So this is known as the transmethylation reaction. So what is the importance of transmethylation reaction? It leads to a synthesis of many compounds which accepts the methyl group from the active methionine, that is S-adenosyl methionine, okay. So this is the importance of transmethylation reaction. So when we talk about this, we should next go on to how the ammonia or the toxic ammonia is transported in our body. So how is it? The transport of free ammonia from the extrahepatic tissues and in the muscle to the liver is in the two forms, okay. That is the glutamine and alanine. So the free ammonia combines with glutamic acid in the presence of glutamate synthetase to form glutamine. Okay, this glutamine is carried to the liver where it gives glutamate and ammonia. Now what happens in the muscle? The alanine transports the ammonia in the non-toxic form by glucose alanine cycle that takes place in the muscle tissue, right? So we'll just have a look at what happens from the muscles. That is a muscle protein. The amino acids are synthesized, that is the branched chain amino acids. The amino group by transamination reaction gets converted to glutamate, okay. Then this glutamate in the presence of alanine amino transferase gets converted to alpha ketoglutarate, pyruvate and alanine is formed. This alanine from the muscle tissue traps in the ammonia and gets and transfers into the liver tissue, right? Again here by the transamination reaction, the pyruvate is given off and glutamate and ammonia, and ammonia is trapped in the urea, okay, and alpha ketoglutarate is also formed here, and this pyruvate by gluconeogenesis leads to formation of glucose. This glucose enters into the blood circulation and enters into the muscle whenever it is required, okay. Then again glucose by glycolysis gets converted to pyruvate and the whole cycle is again repeated, okay. So this is the transport of ammonia from the muscle to the liver by glucose alanine cycle, right? So now when we talk about how ammonia is trapped, next you have to also know the metabolism of ammonia, okay. So what is it? The formation and the metabolic fate of ammonia in the body, right? So what is it? Free from amino acids via the transamination and the deamination reactions, ammonia is formed. And then the degradation of the biogenic amines, ammonia is formed. Then from the amino group of the purines and the pyrimidines also ammonia is formed. And also by the action of the intestinal bacteria urease on urea, ammonia is formed. Now how is it utilized? So first, the foremost, it gets converted to urea, okay. Then synthesis of the non-essential amino acids, also in the formation of purines and pyrimidines. Also it helps in the maintenance of acid-base balance by ammonium ions. It leads to the synthesis of glutamine. Glutamine is a transport form of glutamate, okay. Then you have the formation of aminos, sugars, right? So then what happens when ammonia is formed? It is being utilized. When there is excess of ammonia, what happens we should know, right? So that is called as a ammonia toxicity. So when we talk about ammonia toxicity, the transport of ammonia occurs in the form of glutamine, right? Or as alanine, and never as a free ammonia. So the ammonia is absorbed from the intestine, from the portal blood. So the liver removes the excess of the ammonia as urea, right? So what happens when do we say that there is toxicity of ammonia? So whenever there is increased ammonia, okay, so what happens? The alpha ketoglutarate is not formed by the transamination reaction. So what happens when alpha ketoglutarate is not combining with ammonia? What happens? The alpha ketoglutarate, it enters into the TCA cycle and there is energy released. So this energy is not released because alpha ketoglutarate substrate doesn't enter the TCA cycle. Okay, so because of this there is decreased ATP. Okay, then this is one of the reasons. Then one more reason could be because the glutamate is not synthesized. So when glutamate is not synthesized, there is less conversion of gamma amino glutamic acid, which is nothing but a neurotransmitter, or the biogenic amine that is synthesized. And then another one thing could be because of decreased serotonin levels, the tryptophan metabolism, the ammonia again there will be toxicity. So because of all this, this will lead to the neurological symptoms in the patient whenever there is increased ammonia levels, right? So what is it? So when we talk about ammonia toxicity, we call it as hyperammonemia. So increased, generally the blood serum levels of ammonia is less than 50 micrograms per deciliter. So this we estimate in the arterial blood. So hyperammonemia, we can classify it as into acquired hyperammonemia and inherited hyperammonemia. So when we say it is acquired hyperammonemia, it is seen in cirrhosis of liver, right? Because of the liver development of collateral circulation, and this decreases the portal blood circulation, okay? And then you have the inherited hyperammonemia, that is defect in the enzymes of the urea cycle. There is flapping tremors, slurring of speech, blurring of vision, and death of the patient eventually. So most of the time ammonia toxicity will have the neurological symptoms, that is slurring of speech, tremors, and blurring of vision. Okay, so this is about the ammonia toxicity. So the most of the time the questions that are asked for you people is, they can ask transamination reactions, along with it the clinical importance of all the enzymes, right? And then you have the deamination, and then you can have sometimes questions of can come as ammonia toxicity. So this finishes with the general reactions of protein metabolism. In the next class we'll go on to the urea cycle, which is another long note for you people. Okay, so thank you for listening and hope to see you in the next class.